So again, welcome everyone to track one, where we'll talk a lot more in depth about the data and technology that we use internally at Elucidata and with our collaborators. So in the next 20 or so minutes, I'm going to spend time talking about a framework for defining quality of biomedical molecular data. We'll talk about what biomedical molecular data means, because the term itself can be very broad, as Shweta mentioned in the previous talk. It can mean many different things, but we are going to talk about quality in context of a specific type of data, which I'll define. And then before going into the framework, I would like to set some context of why it's really important to have a common language and a common framework to talk about data quality. And then we'll do a deep dive into types of data quality and a framework that we have found very, very useful when we think about data for any project. And then I'll close with talking about examples of how we can use the quality framework in our work. The biomedical molecular data that I will be talking about today refers to any data that are obtained from biological samples, whether in vivo or in vitro, and they include some measurements made on those samples using any type of technology, whether that's high throughput or low throughput technology. And examples include all types of omics data, all biochemical and phenotypic assays that are often done, imaging, experiments that are done, structural data that are mentioned, that are measured on biomolecules and so on. So for the purpose of our discussion today, biomedical molecular data will mean anything that has some measurements made on biological samples and some information about those samples, which is what we typically refer to as metadata in an experiment like this. Before we go into framework and what data quality means, I just want to again set context for why this conversation is important. So for most of the history of science and research, data have been localized and knowledge was what was widely shared. So often the data were generated, stored, and consumed by the same group of people in physically the same location. An example here is many of you hopefully will recognize this image from the discovery of structure of DNA. Most of the work around the project was done at King's College, and again within a specific research group amongst the same people who were interested in this particular problem. And once the data were generated, analyzed, the knowledge from it was what was widely shared. And of course, this still happens today. Knowledge is still very, very widely shared in terms of conference talks, publications, a lot of literature that's published. But what has really changed in the last 2 to 3 decades is that in research projects, data have taken an independent life of their own. They're no longer just localized in one place, and same data are often used by many, many different people to generate new knowledge, and in ways that are often not even thought of or intended by the original generators of the data. So the example on the screen you see is from the Cancer Genome Atlas, where data were generated by 20 different institutions and different labs in those institutions. And even though every project may not be as distributed as this, if you just look around the data that you may use in your work day to day, that may come from many, many different sources. That may be coming from a CRO that is actually doing the experiment, that may be coming from a colleague who has done the data, that may be coming from another department within your organization that is located somewhere else. So often in projects today, data generation is distributed. And because of the volume and the variety of data that we are generating today, we require different teams to store and manage these data. Again, the example here is from the Genomics Data Common Data Portal, where the Cancer Genome Atlas and some of the other projects are hosted. But this is also true internally in organizations where to store, manage, and archive all this data so that other teams can use it. And then, of course, the data consumption of all of this data then is consumed by independent users who use it in ways that you may not have thought of before. And a consequence of this is that a consequence of data having a life of its own is that there are multiple stakeholders for data at different points in data's life. There are data producers who generate the data. There are data custodians and managers who typically manage and store and make sure that it can be used by other users, and then there are data consumers who then use this data. And often these people don't really talk to each other. And so it's really, really important that there is a shared understanding of what data quality means, and there is a shared language that all of these different stakeholders also speak when talking to each other. Because data quality is a vague term. Often people may mean very different things. So when I say data quality, I may have something else in my mind, versus if somebody else says data quality, they may be thinking of something else. So we really need a common language and a common understanding of what data quality means, so that we can define it and we can improve it for our projects. So a framework that we have found very useful in the projects that we have done and on our platform Poly is to think of data quality as being made up of two components, intrinsic and extrinsic. And what intrinsic means is that this is a quality that's inherent to the data. It's often controlled by data producers. So when intrinsic quality is often governed by, for example, how you have defined the experiment, and I'll go into a little bit more detail about what each of these means, and I'll give some examples. So intrinsic data quality is often controlled while generating the data, and it often cannot be improved after data have already been generated. And most people, if they are using data that are generated by somebody else in their work, this is what they will implicitly think about when they think about data quality, whether it has sufficient quality for their use case or not. On the other hand, extrinsic data quality is not about the data itself, but it's related to all of the systems that are used to collect, store, and get access to the data. And extrinsic data quality is something that is controlled and managed by data custodians and managers. So people like us who work with a lot of data, or in your organizations, if there are data teams, often the things that they will really control for are extrinsic data quality. And it can be improved through the process of data curation, which again, I'll talk about in more detail later. And high extrinsic data quality makes data not just more usable, but it also makes it easier to detect issues with intrinsic data quality. So thinking about intrinsic data quality first, it's helpful to think of intrinsic data quality as belonging to one of three categories for biomedical molecular data. So thinking of experimental design, how the experiment has been designed, and some representative metrics are given below. So we often think about how many total number of samples have been used in a study that I want to use for my work. Are appropriate positive and negative controls included in the study? What is the number of biological and technical replicates that have been used in a condition? Has the experiment been designed well enough so that the effect of a variable that I'm studying can actually be measured, or if there are confounding factors that are present or not? And then the second category is metadata itself. Are all necessary metadata recorded, and have they been labeled correctly or not? And then the third category is the measurement, where we worry about the quality of the measurements that have been made. And again, if it's an omics experiment, we worry about how the sequencing was done and what was the quality of sequencing. If it's an imaging experiment, we worry about what is the quality of the image that we have taken. And here we think of examples like what is the variance in technical or biological replicates or instrument standards that we have used? Are there any technical or batch artifacts in the data? And what does the measurement of my experimental samples looks like compared to controls? Again, these are representative examples. There can be many more criteria within measurement, but the idea is that these are intrinsic to the data, and they can only be controlled while the data are being generated, and they cannot be improved upon once the data have already been generated. So very simple example on your screen, something that will be very obvious to most of us who work with any kind of data, is an experiment done on the left where there are mice of different ages were taken, and hippocampus in the brain of these mice was measured, and each group consisted of 2 male and 2 female mice, compared to an experiment on the right where it's the same experiment design, but the number of samples is different. So each experimental category consists of 16 samples instead of a total of 4 samples. And this will be obvious, but I'll state it because it's important to show that this really affects the intrinsic quality of the data that were generated. So if both these experiments are done, all the metadata recorded the same way, all the measurements are made exactly the same way, even then there will be a difference in the quality of data that's generated, as you can see on the plot here, which is showing distribution of p-values if we do a differential expression of a particular lipid between 2 experimental groups. And if you have more number of samples in your experiment, of course you have more statistical power to differentiate and really isolate the biological effect. Another very simple example, which will appear obvious, is if you do an experiment design like the one on the left, where in treatment 1, all male mice have been used, in treatment arm 2, all female mice have been used, and compare it to another experiment where the genders are equally distributed between the 2 treatment groups. It may be obvious, but the data, if everything else was exactly the same, that the data that come out of experiment on the left will be intrinsically different, will have a different quality than the ones that come out on the right, because in experiment 1, we will not be able to differentiate the effect of gender from the effect of the treatment. And often data like these are weeded out at very early stages, but these are examples that to demonstrate that these metrics really affect the quality of the data. Often these differences will not be as stark as I'm showing you. They will be subtle, but they are important to think about. A third example, again, an extreme example. So given a very well-defined experiment with all metadata recorded, it's important to look at the quality of the measurements themselves. And here it's a Western blot, and sometimes they don't work, and measurements don't work, and data cannot be trusted, compared to one on the right where the measurements actually work and the desired effect can actually be seen. So again, this is an extreme example to show that intrinsically the quality of measurements are different in these 2 cases, even if everything else was the same. But we often see this in subtle ways in data that we are working with. And this is something that we need to think about when we are worrying about data-centric approaches or improving the quality of our data later on. Now coming to extrinsic data quality, which is not inherent to the data itself, but it's related to how data are stored, managed, and accessed by different users. And you will often see terms used to describe this extrinsic data quality as the data are not complete, or the data are not correct, or believability. Are the data believable? Are the data consistent? And you may have used these terms yourself to describe some of the data that you were looking at. And there is actually a term for these metrics of extrinsic data quality, and they are often referred to as dimensions of data quality. And there are hundreds of these dimensions. but often they are related to one of three things. They are either contextual, so you are measuring the quality in context of something specific that you're working on, for example, timeliness or completeness in terms of information that you need. It may be representational, which means it may be related to how data are represented or displayed to users. So if you are looking at an internal database or a public database, consistency or standardization of the data that you see as a user is an example of representational extrinsic data quality. And these can also be related to accessibility of the data, which are associated with systems through which we access data. Examples include findability or uniqueness or persistence of a link to a data. And one of the most well-known examples of extrinsic data quality are FAIR data principles. So the principle of findability, interoperability, accessibility, and reusability are really metrics that improve extrinsic data quality and not the intrinsic data quality. Now, one thing I really want to stress here is, as vague as these terms may seem, they can often be defined very, very precisely in context of a particular data source. So as an example, again, I'm showing simple examples where I'm really isolating what each of these dimensions may mean. So as an example, I can define completeness as the presence of a biological replicate identifier in a study. And if this is my definition of completeness, then data on the left do not meet my quality criteria, but data on the right do. If my completeness criteria was something else, for example, presence of a sample identifier, then both of these data will meet that quality criteria. Similarly, we can think about standardization. If we define standardization as disease values that correspond to unique mesh concepts, then a data is presented on the left where the disease is written in a short form, for example, AD and CD, can mean something else as well, will not meet that, that quality criteria, and the data on the right, where the term is spelled out and uses a defined ontology, will meet that quality criteria. And similarly, standardization can also be defined in terms of data schema, which means that how are the metadata stored? So in this case, I'm defining them as standardization of the name of the field where a specific type of metadata is stored. For example, sample identifier and disease. So if this is my definition, then the data on the right where the names of the fields where metadata are stored have been standardized meets that quality criteria, but the one on the right does not, because I'm using very generic field names to record my metadata values. So this is very briefly about what intrinsic and extrinsic data quality means. Now, what is really important is how can you use this in your own work? So what this framework does is this framework gives us a language to talk about data quality. So if I, as a data manager or a person who thinks about how to store and provide data to different users, I'm talking about data quality, I can be on the same page with a computational biologist who's thinking about using this data. And the way we can use this in our projects is to really think about what do we want from the data in that project. So if we think of data as having intrinsic and extrinsic quality, then we really need to think about if we are in control of generating the data, then we should really think about the complete life cycle of how to make the data as high quality as possible. We should really think about experimental design. We should think about what metadata we are recording. We should think about controlling for quality measurements. And again, a lot of data generators do this implicitly. This is what they spend time doing. But this is often not explicitly talked about. So if you are in control of data generation, think about both intrinsic quality of the data that you're generating, as well as what you can do to improve once that data has been generated. So how are you storing and annotating the metadata? Do you need to reprocess the measurements that you have made to remove technical or batch artifacts? Like, for example, do you need to sample outlier detection? And do you need to structure the data to make it more machine readable, for example? So that's one way in which this framework can be used. And this is, I'll just go back to the slide. This is something we do, we think about a lot on Poly when we are working with data from many, many different sources. We pay a lot of attention to metadata annotation and data structuring. And one of the ways in which we use it is we take data from a source, and this source could be a proprietary data set, a public data set, or a premium data set, as Shwetab mentioned in his conversation. We improve metadata annotation through use of ontologies and control vocabularies. We standardize the metadata schema, and we do on-demand curation for particular use cases. And what that means is that metadata, like the one that you see on your screen, where and again, this is a part of a data set from Gene Expression Omnibus, where the information about gender is given in a column called characteristic CH1, very generic name. The information about liver is given in another column called characteristic CH1_1, and about disease is given in another vaguely defined column. And what we do on Poly is to curate this information. So we standardize the schema to make sure that tissue is always captured in a field that reflects that it's tissue data. Disease is always mentioned in a field that reflects that it's disease. And we do some on-demand curation so that these files are machine readable and so on. And my colleague Shashank is going to talk about this in a lot more depth about what we do here, how we do this curation, and how do we do this curation at scale. Now, a second really important use case that I want to highlight where you can use the quality framework is to bring data centricity to ML projects that you're doing. And what that means is, in a typical ML project lifecycle where you start by defining your use case, choosing the data, training the model, and then deploying the model and making predictions or generating new knowledge. Often, most of the time is spent, or most of the time is actually spent on data, but most of the discussion spans training the model. So knowing that quality of data is critical, and there is a spectrum to quality of data, and some parts of it can be improved. It enables us to ask the question, how can I find the best data for my use case, rather than how can I build the best model for my use case. And so another thing that's really critical here is that often ML projects themselves are made up of people with different expertise. So they may consist of clinicians or biologists who understand the use case really well, and they may consist of data scientists or computational biologists that are really experts in the machine learning side of things. And what quality framework does is to provide a language so that these two groups can really talk about the data itself and not just the model. And what that looks like in practice and how you should implement this is, before you do anything else, at the very beginning of the project, we should really be talking about what are the quality criteria that I'm going to set to choose the data, to choose the highest quality data possible for our model. Again, this doesn't happen as often as it should. Often we look at the potential data pool and say, hey, how can I increase the amount of data that I can send to my model? And what I'm proposing here is that instead of asking, how can I send the maximum amount of data as possible to my model, how do I send the highest quality data as high quality as possible to my model. And the way to do this is to really define and explicitly and deliberately lay out what are the quality criteria that you want to use for your use case. And what that may look like is, for example, if your use case is to predict gene expression signatures in patients with non-alcoholic fatty liver disease, and you have a whole set of data. As an example, let's say you have 100 data that you can potentially use. Instead of just sending all of them and trying to then iterate on the model, what we do is we intentionally lay out what our quality criteria are. What are the data that we actually want to send into the model? And here are some representative ones. These are not exhaustive, but you should think about what are the intrinsic quality of the data that I want to send into my model. So for example, I only want to include data from patients that have been clinically diagnosed with either non-alcoholic fatty liver disease or steatohepatitis. I only want studies that measure both disease and healthy samples from liver. I want studies that only have 3 samples per group, and I want studies that do either RNA-seq or microarray experiments with at least 1,000 measurements, for example. And again, this is a representative set. Your criteria will depend on your use case, but it's really, really important to define those quality criteria at the beginning of a project. And then not sending all of the data that you can possibly find to train your model, but really selecting what is the best data to send to that model. And this would mean not just checking whether which of these data sets meet the criteria. It will also mean thinking about what you can improve in these data so you can send as much, you can send as high quality a data as possible. So this brings me to the end, where I just want to reiterate that data quality improvement is not a one-time done and dusted thing. It's a process, and every single stakeholder in the life of data has a part to play. So if you are a data producer and you're generating data for any project, you should really think about the intrinsic quality of the data that you're generating. And you should think about all the possible use cases that the data may be used for in the future, and generate data with as high an intrinsic quality as possible at the time, given the resources. Then data custodians and managers, people like us, Elucidata, or data teams in your organization, really need to think about defining, measuring, and improving extrinsic quality dimensions. And then data consumers have to be very explicit and deliberate about identifying what are the quality criteria that they are using. Because all of us use these criteria in almost everything we do, but we are often not very deliberate or intentional about them. And of course, at the end, all of these stakeholders need to talk to each other more often than is currently done. So we need to establish a dialogue between different data stakeholders, and we need to continually iterate and improve on processes to improve data quality. Because again, this is not, data quality is not just a one-time done and dusted thing that one person can solve. It's a process that involves people, it's a process that involves systems, and it's a process that involves automations. And with that, I'll close and take any questions.